A phospholipid consists of two regions, the hydrophobic tail and a hydrophilic head. The two hydrophobic tails are made of fatty acids, while the hydrophilic head is made out of a phosphate group. Due to the structure of the phospholipid, the plasma membrane is arranged in a way where heads face aqueous parts and tails face into each other to form a lipid bilayer. The fluid mosaic model states that the plasma membrane is made up of a mosaic of components such as phospholipids, cholesterol, and proteins that move freely and fluidly in the plane of the membrane. In other words, the plasma membrane is nothing but phospholipids and proteins sliding past each other. The fluidity of the plasma membrane means that molecules can go through the flexible membrane and the membrane will still retain its shape afterward. The carbohydrates and proteins act as ID badges that allow cells to recognize each other. The fluid mosaic model has helped explain how membranes regulate the cell's molecular traffic. In the plasma membrane, there are two categories of membrane proteins, integral and peripheral. Integral proteins are proteins which are embedded in the membrane. They have at least one hydrophobic region that anchors them to the hydrophobic core of the phospholipid bilayer. Some only stick halfway through the membrane, while others extend through the membrane. These proteins are called transmembrane proteins. On the other hand, peripheral proteins are found on the outside and inside of the cellular membrane and are loosely attached to the hydrophobic core. Nonpolar molecules such as hydrocarbons are hydrophobic and can therefore cross a lipid bilayer easily. Polar molecules such as glucose and other sugars pass slowly because they are polar along with water. Charged molecules have the hardest time. Although proteins cannot pass freely through the membrane, channel proteins have a hydrophilic channel that molecules can use to pass through. Water is able to pass through the membrane because of aquaporins, which channel water. They can channel up to 3 billion water molecules per second. Cell walls enable cells to maintain water balance better than those without cell walls, but both lose water in hypertonic environments. There are two ways a cell can transport molecules in and out: active transport and passive transport. Passive transport is the transport of molecules or particles across a membrane without the use of energy. There are three types of passive transport: osmosis, diffusion, and facilitated diffusion. Osmosis is the diffusion of free water across a selectively permeable membrane. Diffusion is the movement of particles of any substance so that they spread out in available space from more concentrated to less concentrated. Facilitated diffusion is the selective diffusion of molecules using specific transport proteins along the concentration gradient. On the other hand, active transport is the movement of molecules into a region of high concentration through the use of enzymes against the concentration gradient. Energy required for this type of transport is ATP. The sodium-potassium pump is one of the most important pumps in animal cells, transporting sodium out and potassium in. This pump is especially important as it maintains cell voltage. ATPase is another very important active transport, converting ATP to ADP and releasing energy for later use. Not all molecules are equal, and not all molecules can permeate through the membrane. Cells use different mechanisms to transport these molecules in and out of the cell. Endocytosis is the movement of large molecules into the cell. In endocytosis, the cell membrane surrounds the molecule and forms a phagocyte. Exocytosis is the process of moving a large molecule out of a cell. The cell membrane that formed the phagocyte reattaches to the cell membrane. Phagocytosis is when a cell takes in a solid molecule. Pinocytosis is when a cell takes in a fluid molecule. However, moving large molecules in and out of cells always requires an energy input.